In this video, we will be talking about polysaccharides, right? We know that monosaccharides are essentially, molecular formulas that are some multiple of CH2O, and disaccharides are two monosaccharides linked together by a glycosidic linkage, all found in the previous video. What are polysaccharides? Essentially, polysaccharides are macromolecules. Anytime the term macromolecule is used, it can be inferred that this molecule is quite large. For polysaccharides, this can range anywhere from 100 to 1,000 plus monosaccharides that are linked by glycosidic linkages. And also, they provide structure and storage for both plants and animals, right? And we're going to be going into these examples now. So first, let's talk about storage polysaccharides, right? Storage polysaccharides essentially are used and hydrolyzed or broken down whenever plants or animals need sugar. So in plants, this storage polysaccharide is known as starch. For animals, this type of storage polysaccharides is known as glycogen, right? And glycogen is found usually in the liver or muscle cells of the human body. Next, we have, polysaccharides that provide structure, right? These provide structure and security to the cells. So let's go over the structural polysaccharides, right? For plants, that would be cellulose. And for animals, that will be chitin. Now, glycogen is pretty, it's not as interesting as the other three here: starch, cellulose, and chitin. So for this video, we'll be going over the 3 out of these 4 examples here. And we're going to first start here with starch. So starch essentially is composed of glucose monomers, right? If we forgot how a glucose, looks like, it's something like a hexagonal shape. You have an OH group here, a hydrogen, another OH group here, and a hydrogen. I have a worked example of this in the previous video where we talked about the different types of reactions, biochemical reactions that can occur. And then we have a CH2OH, and then we have another oxygen here, and then there's a OH and an H. Right? This is generally, the structure of glucose. And so starch is composed of glucose monomers, right? So we know there's going to be a bunch of glucose here linked through dehydration reaction. But one thing to remember is glucose comes in glucose monomers come in 2 types of variation. We have alpha. I can write a little bit better. Alpha and beta, right? There's 2 types of glucose structure. We have an alpha structure and a beta structure. Now, this OH group here, right? We call this the plane of the structure, plane of this ring here, right? This is the plane. And this OH structure here, this hydroxyl group, can be seen below the plane of the ring, right? In starch, these glucose molecules or monomers are composed in a alpha structure. Essentially, what alpha structure means is that they are all composed with OH groups below the plane of the rings. How does that look like? So we're going to take this, we're going to just assume that everything here stays the same, and we're only going to look at this region here. So let's just say that we have an OH, hydrogen, right? And then we have another H, right? And then we have another OH, right? This would be linked in this manner. We have a O here, and then we have an O here, right? There was an H2O taken from here, right? 2 hydrogen, 1 oxygen, leaving only 1 oxygen left. That causes this link. This is an alpha configuration. This is what we call alpha configuration, right? And so these structures are all going to be lined like so. They're all going to be lined below the plane. Now, what happens when something is in beta configuration? Beta configuration, so I'm going to write here that this alpha structure is glucose, right? What about beta? Now, beta configuration is when two different monomers are linked, but they are the opposite of one another. Now, what do I mean by that? Essentially, all the glucose monomers that are composed in beta configuration are upside down with respect to its neighbors. Now, what does that even mean, right? Let's illustrate this once again. Let's just say that there is, once again, we know that there is an H and OH, right? I'm gonna draw this out here just so we're clear with how this is structured. We have a CH2OH. I have an oxygen here, then we have an OH, and then an H. If we were to say that glucose monomers are composed in beta configuration, all that would mean is that now it has been flipped. The structures have been flipped. So what that would look like essentially now is, let's just say the CH2O group is at the top. Now the CH2O OH will be at the bottom, with the oxygen being here, this OH group being on the inside, right? This OH group being up top, right? And this hydrogen link happening like this, right? There'll be oxygen here. These two hydrogens will be canceled. And there'll be a link up top like this. So again, if that is a little bit confusing, I'm going to make this model even simpler. Let's simplify this one more step. We're going to take just the OH group. Let's just look at just the OH group here. If the OH group is in the bottom, we have a link that looks, let's just say the link from the top. We have another here, right? We know that with respect to its neighbors, this OH group is now going to be at the top, which means because of this configuration, we're going to have a link that looks something like this, an O. Now that since this is up top, we need to go back to this position, right? Because we know that in beta configuration, each of these monomers are at upside down to one another. So now we're going to have this OH group on top. Everything else is also getting flipped, but just to make this a little bit simpler, I'm only showing the OH. Now that would mean that it's linked like this, right? And once again, we know that this is certainly going to be linked like this because we know that this is in beta configuration and we're going to have an OH up top. And it's going to be this sort of zigzag, almost like this linear shape here, right? And that is enough to make cellulose a type of polysaccharide that humans cannot digest. So the definition for this is monomers are upside down in respect to its neighbors. All right. And this can be found in cellulose. That, upside down structure is enough for us humans and actually most animals to not be able to hydrolyze or digest cellulose. And essentially as soon as we consume cellulose, we just, poop it out, right? Because enzymes that, digest starch because of its alpha linkages are not able to hydrolyze the beta linkages of cellulose because they're so distinctly different in shape between these two molecules, right? So that's actually something kind of important to understand. Cows, for example, have cellulose digesting bacteria that can break down cellulose, but for us humans, we are not able to break this down. We can break down starch, alpha, linkages, but we're not able to break down beta linkages. Lastly, I think I'll be going over chitin, which can be found in crustaceans, arthropods, insects, also used in the medical setting. And chitin is a type of structural polysaccharide. Right? We look at a bug here, right? Like a bug. We sometimes see that when a bug is, shedding, it releases a type of hard, sort of this cracky exoskeleton, right? Chitin is essentially a carbohydrate that is used to build the exoskeleton, right? The outside shell of these insects here. And when they shed it out, usually, I can draw here in a brown, they're usually brown, right? And these skeletons get left out of their bodies, right? This is composed of chitin, right? They're these hard case that's around the soft part of the insects. And chitin is also used for, surgical threads, for example, right? We have threads that are used to sew like incision wounds and stuff like that, and cause healing. Chitin is also composed of that. So we see this structural polysaccharides used not only in animals, but we have found ways to develop polysaccharides, these structural polysaccharides, for use outside of just natural causes, such as developing it to make strong and flexible surgical threads that decompose after a wound or something else has been healed, right?